Okay. So again, this is our lesson 17, corresponding to chapters 26 in the textbook, and this is on biochemistry. And in many ways, this is like a continuation of organic chemistry. When you're like, well, so what? We have these different functional groups. What does that actually mean about what they do? What do these organic molecules do? Well, that's what this is all about. So we've got 4 questions to deal with. The first of which is this. What is a nucleic acid? What is a nucleic acid? What is a nucleic acid? Now, the most concise way to define this, which that's going to seem strange to you because it's actually a long definition, but the most concise way to define this is as follows. Nucleic acids are complex organic molecules composed of one or more nucleotides. Nucleic acids are complex organic molecules composed of one or more nucleotides. So you may have a question after reading this definition, and what would that question be if you have it? What are nucleotides, right? It's always wonderful when we define something as a molecule that contains this, and we don't know what the this is yet. So a nucleotide is a molecule made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base, which we label A, T, C, or G. And that's not that these bases are those actual letters. That's just what we label these bases. A, T, C, or G. So in your mind, you have something associated with that word sugar, right? You think of sugars as things that are sweet. Talk a little bit about sugars in a moment, in another slide. You know phosphate group, right? That was one of our 6 functional groups we talked about last week, phosphate groups. So you know what that is. You should have a very specific image in your mind associated with phosphate group. And then with these bases, you likely do not have a specific image in your mind, and that's okay. These are really complex organic molecules, but we tend to just label those bases A, T, C, or G. Do any of you know an example of a nucleic acid? DNA, which stands for deoxyribose nucleic acid. So DNA is one of our nucleic acids. Probably the one that we know the most about, and it has deoxyribose as the sugar, so that 5-carbon sugar making up those nucleotides is deoxyribose. And then DNA is double-stranded, so every molecule of DNA has actually 2 strands of DNA bound together. We'll talk a little bit more about this as we continue through this slide. DNA has deoxyribose as the sugar. You know it has a phosphate group. Why? What are nucleic acids? DNA is a nucleic acid, and what are nucleic acids again? Complex organic molecules made up of one or more nucleotides, and every nucleotide has what? Phosphate. Has a phosphate group. Which means DNA, which some of our chromosomes are hundreds of millions of nucleotides long, meaning you have hundreds of millions of phosphate groups in those chromosomes. Does that make sense? Okay. And then, so you know DNA's got phosphate in it and a lot of it. You know it has a 5-carbon sugar because every nucleotide has a 5-carbon sugar. DNA has specifically deoxyribose as its 5-carbon sugar. And then you know it's got these nitrogenous bases. Now, the 2 strands are held together. So DNA is double-stranded. The 2 strands are held together by hydrogen bonds. Now, somebody remind me about hydrogen bonds. Are they really strong bonds or relatively weak bonds? Do you remember? When we talked about, we've got ionic bonds, the bonds that form between a positive ion and a negative ion. They are relatively weak. An individual hydrogen bond is a relatively weak bond, and they are bonds that form between the positive pole of a polar molecule and the negative pole of a polar molecule, right? Talked about water forming hydrogen bonds. And so each individual bond is relatively weak. However, if we have a molecule of DNA that is 100 million nucleotides long, now we know we have at least 100 million hydrogen bonds holding those 2 strands together, right? So while each individual bond might be weak, hundreds of millions of these bonds are pretty strong. Okay, so the 2 strands hold together in such a way that A's are always across the way from T's, because the base A forms hydrogen bonds with the base T. So the base A forms hydrogen bonds with the base T. And what bases are left? If we take A and T out of our list, you have C and G. So what do you imagine C is complementary to?
G.
C is complementary to G. So A's will form hydrogen bonds with T's across on the outer molecule, and G's will form hydrogen bonds with C's on the other strand of the molecule. And this is what holds the 2 strands together. These hydrogen bonds. Okay? And how do you break bonds? Somebody remind me from a previous class that we've had. How can you break a bond? Yeah.
Water.
Sure, you can use water to break a bond. Water, as a polar molecule, will oftentimes break up the bonds between other polar molecules, and especially break hydrogen bonds because the water molecules will interact with that. Good. Any other ways that you remember how to break bonds? Yeah, heat. You input energy into it. And both of those will work well to break down DNA potentially. Now, if you've got a really long DNA molecule, it's probably not going to dissolve in water, which is good because the inside of our nuclei is mostly water, and that's where the DNA resides. Okay, so to answer this question, what is a nucleotide? You could answer it by saying C first point, although what I would suggest that you do is you write something like this. It is a single or a string of nucleotides. It is a single or a string of nucleotides bound together by covalent, and sometimes hydrogen bonds. Bound together by covalent and sometimes hydrogen bonds. So it is a single or string of nucleotides held together by covalent and sometimes hydrogen bonds. Because the sometimes hydrogen bonds is going to remind you that that's only going to be for DNA, where we have double-stranded nucleic acids. Other types of nucleic acids are either just single nucleotides, or they are a nucleotide that just has a single strand, where each individual nucleotide is held together by covalent bonds. All right? Any questions? Couple of key terms. If you could take the time to look up nucleotide. To look up nucleotide and to look up DNA. To look up nucleotide and to look up DNA. And when you look up DNA, it might not be a bad idea to draw a DNA molecule. You do not have to, which is why I am not drawing a DNA molecule for you. But it might be helpful for you to remember what DNA looks like and how it is still a nucleic acid. Where are those individual nucleotides? Because that's really what makes it a nucleic acid, is that it has nucleotides. But it's a whole string of nucleotides, and it's really two strings of nucleotides held together by hydrogen bonds. Right? Make sense? Okay. On to our next complex organic molecule type. And so what I want you to do is, after you write this question, what is a protein? After you write this question of what is a protein, I would like for you to work with those around you. And I would like for you to come up with a list of everything you know about proteins. Where do you get them? What do they do? How do we use them? Why do we need them? Okay? Anything you can come up with about proteins. Sound all right? Take about 90 seconds, maybe 2 minutes if the conversations are going well and I don't feel like cutting them off, to come up with basically everything you know about protein. 90 seconds, starting now. All right, what are some of the things that we know about proteins? Anything?
They're organic, found in...
They are organic molecules. Yep.
They're found in meats.
They are found in meats. You got that right. We do find a lot of protein. Meat is pretty protein dense. It's not the only place to find protein, but it is the most delicious place to find protein. Unless, of course, you really like the sweets, and then you can make some sweets with protein-rich flours like almond flour, or better yet, cricket flour. Yeah. If I made you all cookies out of cricket flour, would you eat them? You would? Okay.
If it tasted good.
Oh, they will taste good. Yeah. What else do we know about protein? So yeah, we find protein in meat. It is an organic molecule. Anything else?
They help build muscle.
They do help build muscle. In order to build additional muscle tissue, you have to create a great deal of protein. A great deal of protein inside of those muscle cells. And in order to do that, you need to have a protein-rich diet. You need to have a protein-rich diet. Good. Anything else? So now you know why meat is protein dense, because our muscles are protein dense, and meat is the muscle of an animal. I don't know if you knew that or not, but meat's the muscle of an animal. Which is why you don't call animals that eat insects, you don't call them carnivores, because they're not really eating just the meat of insects, they're eating the entire insect. So you're eating, meat and just everything. So we call them insectivores. But anyways, that's bonus information. Anything else we know about proteins? Okay. Well, let me answer this question for you and define it for you. And again, this is about the most concise definition that I could think of, which will make you smile because this definition is not very concise. Proteins are complex organic molecules made up of dozens hundreds, or even thousands of amino acids. Proteins are complex organic molecules made up of dozens, hundreds, and sometimes even thousands of amino acids. And so if you'll remember, when we were talking about nucleic acids, a single nucleotide is a nucleic acid, right? Remember we defined a nucleic acid as one or more nucleotides? Proteins, you have to have multiple amino acids, dozens of them, before you could ever have a protein. So it's a little different, right? Nucleic acids, even the building blocks of nucleic acids, nucleotides, are still nucleic acids. But the building blocks of proteins are not proteins. Amino acids are not proteins. They are used to build proteins. They are used to build proteins. Okay? Now, proteins are translated from RNA, which is another type of nucleic acid. So DNA is a nucleic acid that never leaves the nucleus. Proteins are translated from a different type of nucleic acid that carries that information out of the nucleus of the cell. Okay, so proteins are translated from RNA that has carried the blueprint for making that protein outside of the nucleus of the cell. And if this were a biology class, I would show you how that works. Even though it's all based on chemical reactions, it's more than what you need to know. So from this point, you can see the relationship between nucleic acids and proteins. You cannot build proteins without nucleic acids. Now, they are a different type of organic molecule, but you cannot have proteins without nucleic acids. Okay? They are both complex organic molecules, but the existence of proteins depends on the existence of nucleic acids. Does that make sense? Okay. Now, again, amino acids, they are the building blocks of proteins, but they by themselves are not proteins. Here's what you need to get a protein. So to have a protein, you have to have the amino acids in the right order and in the right three-dimensional shape. So you have to take the amino acids and you have to put them into the proper order, and then you need to take that string of amino acids, now this whole molecule, and you need to fold it into the proper three-dimensional shape. And only then do you have a protein. Only then. When you have taken your amino acids, you have put them in the proper order, and you have folded that entire string of amino acids into the proper three-dimensional shape, only then do you have a protein. And what does this make you think about proteins? They are complex. And it also makes us think that they're doing something, right? If you need the proper three-dimensional shape, it's usually an indication that those are carrying out some task, right? Where the information, it's not just about the information, it's also having that information in the right form. And so many of our proteins, not all of them, and probably not even most of them, but many of our proteins function as enzymes. Many of our proteins function as enzymes, which are biological catalysts. Many of our proteins function as enzymes, and these enzymes are biological catalysts. I'm pretty sure we've seen this word before. Catalyst. Anyone remember where we've seen this word before? It was in one of our labs, and it was in the lab that you just did a write-up for. We use something to act as a biological, or not a biological, we use something to act as a chemical catalyst to ensure that the reaction of hydrogen peroxide turning into oxygen gas happens quickly enough that we create that foam. We create that foam that we needed to create for lab 5, right? And so we use potassium iodide as our catalyst, and it sped up that reaction. This is what enzymes do. They speed up reactions. Joe. Yeah. So these enzymes, as they act as biological catalysts, they take chemical reactions that are happening in your body and they facilitate them. So they happen at a rate that keeps you alive. Because it's not just a matter of making sure the right reactions happen. It's also a matter of making sure that the reactions happen in an amount of time that's conducive to life. Okay, let me give you an example. This is an example beyond this course. You can write it down or not write it down, whichever you like, but I want to explain it to you so you can appreciate this. So our bodies, or our cells, store most of their energy as a molecule called ATP. And that molecule happens to be a nucleic acid, but that's another story for another time. So cells store most of their energy as ATP. And so when a cell needs energy to do something, it typically will cut a bond in ATP and release that energy to facilitate some action. The average cell in your body makes and breaks 100 pounds of ATP every single day. The average cell in your body, every individual cell in your body, well, not every cell, but the average cell in your body, of which there are trillions, makes and breaks 100 pounds of ATP every day. And so it just shows you, so all of the ATP it makes, it breaks, right? Because otherwise you'd be accumulating weight at an astronomical rate. That is a reaction that you can take ATP, and if you put it in water, it will break down, that bond will break, and it will release energy. But without an enzyme, it will take hundreds, if not thousands of years for that reaction to happen. You can't wait around for that, right? If every cell in your body is making and breaking 100 pounds of ATP every day, that is a reaction that is happening about 100 million times every second. About 100 million times every second. And the only way it can happen at that rate is for there to be enzymes that are facilitating that reaction. And these enzymes that are doing that, they're proteins. And those proteins are made from nucleic acids. Nucleic acids are involved in the process of making proteins. Make sense? So what is a protein? Again, you can answer this by writing C first point if you want to, but here's what I would write. I would write that a protein is a properly shaped string of amino acids, a properly shaped string of amino acids that carries out the work of a cell. A protein is a properly shaped string of amino acids that carries out the work of a cell. If you want to write C first point, I wouldn't knock you for writing that, because, I mean, I came up with this definition, so obviously I liked it. But I might write that second statement. All right, a couple of key terms. I would like for you to look up amino acid, and I would like for you to look up enzyme. Amino acid and enzyme. Okay, any questions about this question? We have two more complex organic molecule types to talk about. We've talked about nucleic acids, we've talked about proteins, we've got two more. Did you have a question?
On note, so the ATP or whatever is made from what?
Oh, ATP? Well, you mean like what are the materials to make ATP, or what are the things that facilitate making
I was
ATP?
wondering because it says it has 100 pounds of it, so they're making it out of something. I want to know what that is.
Yeah, they're making it out of material that's already there. Now, you have to understand, if you take, so the pieces that you use to make ATP are ADP and inorganic phosphate, okay? But this is beyond what you need to know. But if you take that, you take an individual molecule of ADP, you take an individual molecule of a phosphate group, and you put them together, you have ATP. Now, if you do that one time, you have one molecule of ATP. But if that same molecule is broken and reassembled 1,000,000 times every second, now you have essentially made and broken that molecule 100 million times, even though it was all built from the same two pieces, right?
Yeah.
And so that's how you can get numbers like that, where it's like, this doesn't make any sense. The cell doesn't have 100 pounds worth of material, because if there are 1 trillion cells in my body, and each of them had 100 pounds of material, that's a lot of weight.
Okay, that's really interesting.
Especially if you have the two same things you're sticking together, like two Lego bricks that you just go.
Yep. Hundreds of millions of times every day. Yep.
It's a lot of work.
Because somebody that weighs less than 100 pounds can't
There's-
have 100
correct.
pounds worth of.
And definitely not in every cell.
Definitely not for
Yeah. Yeah.
Okay.
But it just, the point of hearing that is to just note how many times this reaction has to happen every day inside of the cell. Just billions and billions of times that this reaction of breaking ATP and rebuilding it happens in a cell in a given day. All right, our next question. What is a carbohydrate? What is a carbohydrate? And we hear a lot about low carb diets. I mean, you maybe don't interest yourself with that too much. But what is a carbohydrate? Simply put, carbohydrates are sugars. Sugars are carbohydrates. Carbohydrates are sugars. They are two terms for the same type of organic molecule. Again, carbohydrates are sugars. Sugars are carbohydrates.
Guessing that does mean that how should be the version I think of, because bread is most has a lot of carbohydrates in it, and you don't think of bread as sugars.
Correct, but it is. Yeah, because again, we have a certain understanding with sugar. We hear sugar and we think something sweet. Not all sugars are sweet. The sugars that are sweet are the sugars that are sweet are those that excite the sweet taste buds on your tongue. So a lot of the complex sugars that you find in breads, you don't recognize them as sweet, but they do the same thing for you. Like if you ate a big piece of bread, if you're eating really highly processed sugars like a candy bar or drinking a soda, the amount of sugar in that is really dense, like more than what you would have in bread. But you're ultimately going to get to the same thing if you eat bread versus drinking a soda. Just in the soda, you're getting a lot of sugar. A lot more than what you would get in a slice of bread. I wonder how many slices of bread you would need to eat to get the same amount of sugar as a soda.
About 3.
Now, do you think so?
Yeah.
Yeah.
Depends on how big the
Well, it's not just... Yeah, I believe that. Yeah, somewhere between 3 or 4. Yeah, 3 and 4 slices of bread. Yeah. So another way to define a carbohydrate, so you could just say a carbohydrate is a sugar. Another way you can define this is to say that they are complex organic molecules made up of one or more monosaccharides. You getting this theme that we've got building blocks for making these complex organic molecules? Like with nucleic acids, we have nucleotides. With proteins, we have amino acids. With carbohydrates, we've got monosaccharides. So they are complex organic molecules made up of one or more monosaccharides. So true or false, a single amino acid could be called a protein. That is false. True or false, a single nucleotide could be called a nucleic acid. That is true. True or false, a single monosaccharide could be called a carbohydrate. It's also true. Okay? And so carbohydrates is getting back to like the nucleic acid. Now notice nucleic acids actually have a sugar as part of those nucleotides, right? Every nucleotide has a 5-carbon sugar, which means you take something from this group of complex organic molecules and you use it to build another type of complex organic molecule, right? And then we said proteins can't exist without nucleic acids. Nucleic acids can't exist without sugars because there's sugars in nucleic acids. So proteins can't exist without nucleic acids. Nucleic acids can't exist without sugars. And now you're like, man, there's like this balance where everything is dependent upon everything else. And there really is. There's a really nice balance there. Okay, a little bit more about carbohydrates. Carbohydrates contain 2 of our functional groups that we talked about last week. They contain hydroxyl groups and carbonyl groups. Every sugar, every carbohydrate is going to have at least one hydroxyl group and at least one carbonyl group. Typically, sugars have one carbonyl group and a hydroxyl group for every other carbon. But anyways, that's bonus information. So you tend to have a lot of hydroxyl groups in sugars and one carbonyl group. And you're like, so what, Dr. Engel, you really didn't talk to us much about those functional groups and what they do, but you did tell us that they are they are chemically active, right? Those functional groups make organic molecules more complex and they make them more chemically active. And so these sugars, these are fairly chemically active biomolecules, okay? Which is kind of what makes them wonderful to build things out of. They're also very sticky, and the reason they're sticky is because they're chemically active. They tend to form bonds with other types of molecules. That's what makes them sticky. Okay? That's why sugars tend to be sticky, carbohydrates tend to be sticky. It's just nice. Okay, a little bit more about carbohydrates. The number of carbons varies from sugar to sugar. The number of carbons varies from sugar to sugar. Most monosaccharides have either 5 or 6 carbons. Most monosaccharides have either 5 or 6 carbons. 5-carbon sugars, we call them pentoses. 6-carbon sugars, we call them hexoses. DNA has a specific sugar in it. And what is that sugar again? Deoxyribose. And what type of sugar is that? Is that a 5-carbon sugar or a 6-carbon sugar? It is a 5-carbon sugar, right? We say nucleotides have a 5-carbon sugar. DNA has specifically deoxyribose. And so we would call that a pentose, that 5-carbon sugar. And again, you can't have proteins without nucleic acids. You cannot have nucleic acids without carbohydrates. You could have carbohydrates without either, but you can't have the other two without carbohydrates. Okay. So, to define this, again, you could write C first point. You could write C second point. You could write C first 2 points. You could write C list. Or you could write this, that a carbohydrate is a monosaccharide or a string of monosaccharides. A carbohydrate is a monosaccharide or a string of monosaccharides. And then as far as key terms, the only key term I would like for you to look up on this slide is monosaccharide. Monosaccharide. One shall be enough. Any questions about this? Okay. Cool. Our last question that we're going to deal with. What is a lipid? What is a lipid? What is a lipid? So lipids are complex organic molecules made up of typically only hydrocarbons. So lipids are complex organic molecules typically made up of just hydrocarbons. Lipids are complex organic molecules that are typically made up of just hydrocarbons. And you're like, how could they be complex organic molecules if it's only made out of the simplest of our organic molecule types? And the answer is it's because it's a bunch of these pieced together. Now, somebody remind me about hydrocarbons. What are they made of? Just hydrogen and carbon. And what do we know about hydrogen and carbon as far as how they share electrons? Yeah, Joe.
The hydrogen only has one electron,
Uh-huh.
as another,
It does. That's true.
and carbon forms four.
Yeah, 4 covalent bonds. Yep. And we also know that they share electrons evenly, right? They have about the same affinity for electrons, the same ability to attract electrons to their nucleus. And so they share electrons evenly, meaning that they do not make polar bonds, meaning they tend to be nonpolar. Okay? They tend to be nonpolar. They are usually nonpolar, which means that they will not dissolve in water. Okay? They tend to be nonpolar, and they tend not to dissolve well in water. Okay, so knowing that that's the case, what types of materials are going to be lipids? What are some things that you know from either cooking or some other experience that you've had that you know does not dissolve well in water?
Oil.
Oils? Good. Yep, so have you ever had Italian dressing? And you have to shake it up before you use it because the oil layer separates from the water layer? Milk, as it goes bad, it becomes no longer homogenized and the fat layer will separate from the water soluble layer. Have you ever had butter on your hands and tried to wash it off with water? And it's very, very difficult until you start to use some soaps. And soaps are interesting because soaps are both polar and nonpolar. Soaps are designed to where they'll interact with nonpolar materials like the butter on your hands, and they'll also interact with the polar materials like the water you're using to wash the soap off, and it will wash nonpolar materials off your hands. It's really nice, okay? And so those kinds of things are lipids. So lipids can be fats, they can be oils, they can be waxes, and they can even be a category of materials that we call hormones. Okay? And hormones are ways of using chemical signaling in your bodies. And so you've got hormones that are associated with secondary developmental characteristics. You have hormones that are associated with your immediate response to something like adrenaline, epinephrine. Okay? You've got hormones that are used in cell signaling. Now lipids, they don't dissolve well in water, but they are a great way to store energy. They're simple, relatively chemically boring organic molecules well, they're still complex, but they're more simple than some of our others. But they are a great way to store energy. They are very energy dense. And so, pound for pound, fat provides you the most energy in your food sources. Okay? Pound for pound, fat provides you the most energy of all of these complex organic molecule types. Okay? Great way to store energy, fairly chemically boring and unreactive. Okay? Which is nice. That makes it another nice way to store energy that way, because it's not going to tend to deteriorate until you do so purposely. Okay? So it becomes a great way to store and to process energy. All right? Any questions about this? I don't have any key terms for you, but if you want to answer this one, what is a lipid? You could say C first point, or you can say that it is a nonpolar organic molecule made up primarily of hydrocarbons. It is a nonpolar organic molecule made up primarily of hydrocarbons. Yeah, Tom.
I guess this probably isn't how it works, but when it says it's a great way to store energy, does that
Uh-huh.
include partly why heat is transferred so well in oil? I guess it's probably not, but
No.
I don't know.
No. Although the nice thing about oil is a lot of oils can get to a really high temperature before they begin to boil, and a lot of oils can get to a really high temperature before they start to burn and kind of develop like a really bitter taste. Yeah.
You can address that answer.
Yeah, so they are nonpolar organic molecules made up primarily of hydrocarbons. Yeah, nonpolar organic molecules made up primarily of hydrocarbons. Right. So I will